So for lab today, we will discuss about the differential test 1. And this is the list of lab exercises that we will do. The first one is the phenol red broth, or called PR broth, or PR tubes. We have the bioascalin test, we have the starch hydrolysis test, and we have the blood agar plate. We would do this as a team per pod, and then you guys would split the work, and each person could execute one of those lab exercises. Now let's talk about the phenol red broth, or the PR tubes. So this is a differential medium only, which means that all types of bacteria can grow in here. But once they grow, we are going to learn something about them. On a regular medium, if the bacteria is able to ferment the sugar that's present, usually it's going to be glucose, we can't tell if they're actually fermenting or not. So when we inoculate the bacteria in a PR broth, it's because we want to detect fermentation of different sugars, okay? So with the fermentation of sugar, we may see acid production only because it changes the pH of the media, which means that the pH drops, so we see a color change, or we can see a combination of acid production and gas formation, so we can see both as well. So which sugars are we trying to observe fermentation? Would be glucose, sucrose, and lactose. So all those sugars are differential ingredients, because if the microbe is able to ferment one or more of those sugars, we can see a color change. How are we going to see a color change? Because we have a pH indicator. So the color of the broth wouldn't be brownish as we usually have for broth. This broth, they are red. They look red because there is phenol red mixed in the broth, and phenol red is actually red at neutral pH, because that's how we start with the medium, in about neutral pH as would be inside of our body, because that's what microbes like, the ones that we work in the lab. So it has to be about 7.2, 7.4. Okay, but the thing is, phenol red turns yellow if the pH drops, if it becomes very acidic. And if the pH actually rises, it will turn pink. Okay, so if we have an increase in the pH, that means protein degradation, because the bacteria may not even like any of the sugars, and then they might use the proteins that we call peptones. They'll be present in the medium. Okay, this is how we do this. When you guys would go back, you go in the back of the room to do some shopping, you guys would grab 4 tubes that had glucose on it, 4 other tubes that have sucrose on them, and then another 4 other tubes that had lactose on them. So all the tubes would be color coded. So the tubes that had the red cap would be for glucose, the green cap would be for sucrose, and then the yellow cap would be for lactose. Okay, and then we would all label the tubes properly, because each sugar that has a different, each tube that has a different sugar would have to be inoculated with one of those 3 species. Okay, so we would inoculate with E. coli, A. faecalis, and P. stewarti. And then we also do a control. So a control in this case would mean no bacteria whatsoever, no inoculation. Okay, so basically you would need to do, you guys could split the work to do this quicker. Okay, so each person could grab 1 bacteria, and then let's say if you are the one doing E. coli, so you'd have a glucose tube, a sucrose tube, a lactose tube, and then you would aseptically inoculate E. coli in each of them. Okay, so each bacteria would have a set of 3 tubes, 1 for each sugar. And then we would separate on the side one tube for each sugar to be our negative control. So we wouldn't even open those tubes. We only write control on them. That's all we'll do. Okay. So one thing in particular about these tubes is that you guys would notice that inside of the big tube, there would be a smaller tube. So there's a glass tube that it's actually upside down, and there's an opening in here. And this tube is called Duhan tube. And the only reason we have a smaller tube upside down inside of the bigger tube is because if there is fermentation with gas production, the gas would be trapped inside of the tube, because naturally when the bacteria ferments, the gas stays on the top and then eventually it leaks out of the tube because the caps are loose. There's no way we can notice if there was fermentation except for the smell, but that's not enough. Okay, so we would actually have to have a visual indication of that gas formation. Then it would move to the next lab, which is called bioascalene test. So this is the only medium today that would be selective and differential. Okay, and this medium it's used to isolate and identify bacteria that's able to break it down, hydrolyze ascoline. So ascoline, it's a compound made of glucose and scolatine. And then one thing that certain microbes and certain bacteria can do is to break down this compound, ascoline, in the presence of biosalts. And here, usually we have a certain group of bacteria called the group D streptococci and enterococci that have this very unique ability to break down ascoline, this compound made of glucose and scolatine, in the presence of biosalts. Okay. And the reason we are trying to see this in the presence of biosalts is because when we think about the different types of bacteria that we have in our small intestine, some of them may not be able to break down certain compounds in the presence of biosalts. And biosalts are released in the very, first part of our small intestine. And that's going to be important to dissolve the fat from our diet. Okay, so because the microbes, they'll be present throughout our entire small intestine, some of them may not even be on that side because they may not be able to break down anything because of the biosalts. But this group of bacteria, they can do that. They can take this compound right here and break it down in the presence of the biosalt. So now for ingredients, we're going to have bio in here. That's also called oxygal. Okay. And then this ingredient will be the selective agent for the group D streptococci and enterococci, which are both gram positive. Okay. So now, out of those organisms that can actually grow in the presence of bio, only a few can actually break down ascoline into, glucose and scolatine in the presence of that compound. Okay, because that's the whole idea. It's not just be there, but be there and also break down this compound, which is going to be ascoline. So now ascoline then is going to be our differential ingredient, because if the bacteria grows, that's fine. They can tolerate the salt, the biosalts in there, but then it's not just that. They have to be able to break down ascoline in the presence of bio. Okay, and then when they do that, we have to find a way to see it, because everything for those differential medium going to be related to visual indication of the results. So we have an indicator here which is not related to pH. It's something called ferric citrate. So this different compound that we have is going to actually react with the byproduct of the breakdown of ascoline. So as I said, ascoline can be broken down into glucose and scolatine. So once scolatine is made, it will react with ferric citrate, and then we would see a black color on the medium. Okay. So this test would be very simple. We would take two slants. So these lens for bio, they look brownish, a little bit much darker than usually we have for regular medium, okay? And the only thing you would need to do for this is, in addition of labeling the tubes, of course, we would inoculate M. luteus and E. faecalis. And E. faecalis is a BSL2. You would have to be careful with this one. But because this is a slant, we only do that, zigzag fishtail pattern on the surface, and that's how we would do. And then we would wait 2 days to get the results. Then we would move on to the next lab, which would be the starch hydrolysis. And this one is a differential medium only, okay? So starch is a very, very complex polysaccharide made of, different glucose molecules. So this bunch of glucose molecules, once they are released from the complex micromolecule, then the glucose can be used to produce ATP, but not as a huge, polysaccharide. That's not going to happen. So the thing is, if the bacteria can actually take starch as a source of sugar, first they have to break it down, because starch is a big polysaccharide. It will not cross the cell membrane. So we talked about transport in the cell membrane before, and most molecules cannot cross. And even the ones that might cross, they might need a channel or pore for that. And that's not the case for starch because it's too big. So what cells, bacterial cells evolve to do, they produce enzymes, and then those enzymes will be released outside of the cells. And then because the starch is present outside of the cells, it can be broken down into smaller sugars, which would be glucose. And now glucose will find a channel to cross the cell membrane. So certain types of bacteria may have alpha amylase or oligo-1,6 glucosidase enzymes, and then if they have both or just one of them, that is enough for them to actually produce it and then release outside of the cells. And then as they break down the starch into simple, glucose molecules, now glucose would have that chance to cross the cell membrane actively because there's ATP involved for that. And then from that, once glucose gets inside of the cells, so now it can be used as a source of energy to produce ATP. So now this is the thing. Once we inoculate, a plate, because this is going to be a plate, we want to see if starch is still present in the medium. So this is a medium that would look, similar to a regular plate as we have, except that there's a starch mixed in the agar. Okay, so once the agar is still melted, and you can mix starch on it, it has to be well mixed. Because otherwise it can give results that we cannot rely on them. So starch has to be well mixed, and then once the plate solidify again, then we can inoculate on the surface. So starch will be our differential ingredient, because technically anything can grow on this plate, but then we'll find out if the bacteria has the enzymes for that. So this lab, we only need one plate. That's all we need. So we would take the bottom of the plate, we would make 3 sections, and then one section we would do a streak line with E. coli, and then another streak line with B. cereus. And then the third section we will do nothing. That will be our negative control. So we would do this aseptically from a slant, so there's no chance of spills or anything. And this would just require one person to do it. So it's super simple. Then our last lab would be the blood agar, which is also differential medium. And, as the name says, there's blood on the plate. So the plate is really red because of the presence of sheep blood on it. And we have done an exercise similar to that already. So, but for this one, instead of trying to inoculate just a pure culture of bacteria into the plate, we would take a throat sample. So we would take, 2 people in each pod, and then we would take a sample from your throat, and then we would streak on the plate. Because we know there are several species of, gram-positive cocci, especially strep and staph, that can actually destroy the red blood cells. So they have hemolytic patterns, and those would be beta hemolysis, alpha hemolysis, and gamma hemolysis. And there are certain species of streptococci then have what we call streptolysins. And the streptolysins, they may have a better activity if they are under anaerobic conditions. So we can actually try to create two environments, aerobic versus anaerobic. And then if you have a strep species on your throat as part of your neuromicrobiota, or maybe because you have an infection going on, we could perhaps identify the presence of strep by looking at the aerobic versus anaerobic conditions that we create. Okay. So this is how we would do this one. Because this is the last exercise, you would have to change your gloves just to make sure that we did not got any contamination on them, just in case. And then we would have to have 2 volunteers to get their throat sample collected, and we'll take a cotton swab. And then our goal would be getting the sample from the tonsil area in the past, so in the back. So this would be very quick as well. And then we would take that cotton swab and do the very first streak on the plate, because for this plate it's best to do a 4-quadrant streak instead of a streak line, okay, or spot inoculation. That's how we call it, streak lines as well. It's better to do a 4-quadrant streak, because remember, this is a type of streak that it's better, it's more appropriate if you have either clinical samples, as is the case for a collection from the throat, or just because you have a sample that has a high number of bacteria. So we have a lot of bacteria in our throat, so it's a combination of the two things right now, because a clinical sample and there's a lot of bacteria. So if you want to get colonies, you have to do a 4-quadrant streak, not a streak line, okay, because that's how you can identify different colonies, and some of them may do hemolysis, some of them may not, because you're not going to have just one bacteria growing on that plate. So then again, we would take that cotton swab and we would do the very first quadrant, and then this one will be easy because it's just from the the cotton swab. And then we would just pose that cotton swab in the biohazard bag just in case, because we don't know what you have in your throat. And then we would finish all the other 3 quadrants using your metal loop, okay? And you guys would know how to do this. You would have to flame in between each quadrant before you go to the next one, and then you'd have to follow that pattern of the 4-quadrant streak. So now, how are we going to create the aerobic versus anaerobic environment? So instead of having two, like a chamber that we have done in the past for create an anaerobic environment, so for this one, we would actually take our loop and we would turn the loop like a little knife, and then we would cut the agar, because what we can do with this procedure, we can create an environment kind of underneath the agar on the surface. Instead of being all the way on the surface, we could create a deeper environment, and then deeper in the agar you'll be low in oxygen. So we would try to do 2 cuts in the center. So you can see here that in the center there's no bacteria. So we will try to do this here first. So after finishing all your 4-quadrant streak, you flame your loop again, and then as the loop is now sterile, you do 2 cuts in the center, and then without having to just flame it again, we will do the same thing now in the first quadrant, because here we know there's a lot of bacteria. That's the first quadrant, so you do the same 2 cuts in here, and then our goal would be to look for that pattern of hemolysis around the cut, because if the bacteria is able to destroy red blood cells, we would see a better destruction under anaerobic conditions if that bacteria was a strep species. Okay, so that's kind of the goal. So eventually, if you had an infection going on that you are aware of, we would still, we'll try to be like even more aware of that and write this on the plate just in case someone could open the plate by accident. Okay, it's always good to be careful. And at the end of this labs, this is what we would have. So for each species of, for the PR broth, we would have a set of 3 tubes plus the controls. Okay, so there'll be like a set of controls for each pod, because we need to look at the color of the controls before we can actually look at each tube that's been inoculated. And then you'd have 2 tubes for the bioascalin test, and you'd have 1 plate for the starch hydrolysis, and then technically would have 2 plates for the blood agar. Okay, 2 patients for this one. And then 2 days later, we would look at the results. Okay, so let's start with our phenol red broth. So you'd have to look in your lab manual, to compare the results first, just to kind of see what the results look like, because this is an example of the PR glucose only. Okay, and this is just showing like the different colors that you can see depending on the bacteria. So the control be here in the center, and then you'd have to compare the color of the control with the other ones that are being inoculated. So this one, for example, because it's very yellow, yellow means it's acid production. So there's fermentation of the glucose in here. Okay, but then you can see inside of the tube, there's no gas at all. But now when we look at this one is even like lighter, yellow, but this still means fermentation. But you can see how much bubbles you got trapped inside of the tube. So this means acid and gas production. Okay. This one over here, there's no reaction compared to the control. Means there's really, the bacteria did nothing. Didn't use any sugar or protein. But one thing that may happen, the bacteria may actually use other sources. They're present in the medium. And then instead of turning yellow or just no color change, they will turn pink. And that means alkaline condition. Okay, because the pH has raised in here. So that's why it'll turn this color. Okay, but now let's look at the results that we would get. Okay, so I have a table over here for you that kind of summarize the expected results, and most of the time we actually get those results. But let's start with the controls. Okay, remember, this is the tubes, they're all color coded. So these tubes, they're in red, those are glucose. Okay, that's the only sugar we have in here. The ones in green, this is going to be sucrose. The other ones in yellow, they're going to be lactose. Okay, so this is the color they would have without any bacteria on them. So they look all the same. Okay, so now if we look at E. coli down here, okay, so E. coli, let's just look at one sugar at a time. So if you look at glucose, you can tell easily that E. coli ferments glucose because now the medium is yellow, and there is also gas formation in there. You can see the bubbles trapped inside of the tube. So that's why we see here E. coli, acid plus gas, for glucose. Now if we look for the sucrose, there's no difference of this color and this color, and that's how you see negative in here. There's nothing going on. Okay, now when you look at lactose, you see the same result as we saw for glucose compared to the control. So that's why you have here acid plus gas. Okay, so now if we look at A. faecalis, so A. faecalis, you're just looking at overall, all those 3 tubes compared to the controls, there's no difference. That's why they're all negative across the table. Okay, now the last species, you can see here that we have fermentation for both glucose and sucrose, but none of them have actually, gas production. And that's how we see just acid here, and there is no difference for, lactose compared to this control. Okay, so this would be the interpretation of these results. So for the unknown project, this is what you would look if you decide to inoculate, this medium. You'd see if there is a color change to yellow, and then in addition to that, you'd look for bubbles inside of that tube. Okay. So now let's look at the bioascalin test. So this is what the control tubes look like. So this medium, it's a little bit brownish, naturally brownish, okay? And then we would inoculate, each tube, one with E. faecalis and then the other one with M. luteus, okay? And then again, if, the bacteria grows in here, it means that, the bacteria there was a color change. It means the bacteria was able to break down ascalin into ascolatine and glucose. And now ascolatine is going to react with ferric citrate and turn it to this black color. So if you see a black color compared to the control, it means that it is positive for the bioascoline. It was actually able to break down ascoline into ascoline and glucose. And then for M. luteus, because it's negative, because it cannot do any of that, we would see the same color as we do with the control. It means no, there's no bacteria in here. But this one, even though there's bacteria on it, there's nothing happening. So we would see the same color in this case. So the only one positive would be E. faecalis. So we would have to make sure that the this black color would be at least like halfway in, the medium, just to make sure it's positive. Now we would look at the starch plate, okay? So this is what it would look like initially. So when I give you guys the plate back, you'd see a lot of growth for E. coli, but way more for B. series, way more. The B. series look like a little tree growing on the plate. And then of course nothing on the control. So right now, the way how the plate is, you can't tell what's going on, okay? Because the thing is, there's a starch as a source of sugar on the plate, but then our goal is to make sure that the bacteria is able to produce enzymes that can break down starch, and then now they can use the glucose, the simple sugar, as a source of energy to produce ATP. But if the bacteria is actually using starch, it means starch would not be present around the growth. So that same spot where the bacteria is growing, starch would not be present anymore because they're breaking down and they're eating that. Okay. So how can we tell if starch is still present on that spot of the plate? We would have to add iodine. So the same iodine that we do that we can use for the gram stain, or usually do like a more concentrated iodine, which is darker. So we add some on the plate and then we wait like a minute or two, and if starch is gone from the plate, we would see this beautiful halo around the growth of the bacteria, which is not the case in here. So this is just the bacteria, of course, but around the bacteria you still see the dark color. So what iodine is doing here, it's actually interact with starch. So every part of the plate that we have starch, it turns into this dark color. But if starch is missing from that spot of the plate, because it has been used, the bacteria actually is eating that starch. So now there's nothing to interact with. So iodine is not going to interact with anything. And then we would see this, beautiful halo around the growth. So B. series is growing way more than E. coli because it's able to use the starch. And that halo showing like no binding of iodine with the starch, it's a very clear result of a positive result for starch hydrolysis, because that's what the bacteria is doing here, breaking down starch using water. That's why we call hydrolysis reaction. Okay, so this result is very straightforward. So now we would look at the blood agar plates. And for this one, we would need to go in the biosafe cabinet in the back of the room, because in case we want to open the plate, we have to do this safely. And also because usually we have controls in the biosafe cabinet to compare what we expect. Okay, so usually we have controls from some of the pure cultures that we keep. Okay, and remember that when we look at blood agar plate, we want to try to identify the pattern of hemolysis, which means destruction of the red blood cells. So if we have a beta hemolysis like we see over here, it means complete destruction. So if we look against the light and would have a particular light box inside of the biosafe cabinet, okay, that's how you could see the pattern. So just looking at the plate without being against the light, you cannot see any pattern. So you have to look against the light. So here we have Streptococcus pyogenes, which can actually do beta hemolysis, so complete destruction. All this clearing means the red blood cells are missing in that part because there's just no red blood cells anymore. But now, if we see what we call alpha hemolysis, it's partial destruction. Now the color of the growth turns a little bit greenish, and that's because it's partial, okay? And then you don't see all that clearing. It's mostly just, greenish. And now if the bacteria is able to grow on the plate, but then they do not destroy any of the red blood cells, then we call that pattern gamma. And the bacteria is grown in here, you can tell, but you don't see any difference in the background compared to what the growth looks like. Okay, so this will be the pattern. But the example that we have here, it's our lab technician's throat culture. Okay, and this example, we are not looking against the light, so we can't tell for sure what pattern of hemolysis we would get. But this is important to also look without being against the light, just to see the different colonies. Okay, so we can see some white columns over here, and we can see even like some greenish color, even though it's not completely against the light. We can still see, different color. Whatever this color is, it's not the same as this one in white. Okay. So as you can see here, this is the quadrant number 1, and then we got isolation, as we move from 1 to 2 to 3, and we didn't get much in 4. You can see the marks of the streak in here, but we don't have any bacteria. But the only way to get isolation from a clinical sample or any other sample that's have a lot of microbes on it, it's doing a 4-quadrant streak. So a streak line, a spot inoculation, it wouldn't be appropriate for this. Even like not a zigzag, it wouldn't be appropriate at all. So we would try to find, colonies. They're kind of different from the other ones, to see if any colony would have more of that hemolysis pattern compared to others. And then I don't know if you guys will be able to see here, but then there'll be like 2 cuts in the center of the plate, and then it would have another 2 cuts over here in the first quadrant. Okay, and then as we look against the light, because that's the part you can't see from this picture, as we look against the light, we would try to see if there was more hemolysis around that cut, because that means the bacteria that got inoculated inside of the cut, because once you make the cut after you already streak the bacteria all over the quadrant number 1. So you'd kind of push down the bacteria with the loop, and then the bacteria that grows under the surface, they could perhaps break down, destroy the red blood cells more than they would do in the surface, because if they are streptococci, they could have streptolysins. So they're more specialized enzymes that they work better at anaerobic conditions. Okay, so from this picture, unfortunately, it's very hard to tell which pattern we would see, but very likely in overall we would see a pattern of alpha hemolysis, because most of us, of course, we all have staph and strep species in our throat. They are part of our normal microbiota. Okay, but most of them would only do alpha hemolysis, so we would expect a greenish color. Very rarely you're gonna find just gamma. Okay, so this is just what I'm telling you, what we would probably get for most of you. Once in a while, we get students that already have some infection going on, even though they don't show any signs and symptoms of sickness, which they may never show, because the immune system, it's taking care of everything. But then it's very that's nice to see the pattern on the plate when we get the results like that from students' throat, just getting like beta hemolysis. Sometimes we get like predominant colonies, they're isolated, and they all have a pattern of beta around them. Instead of being over here, we see them just over here. So but this is just the example that you have. So you're gonna have to work, your results for the data sheet are gonna have to be based on our Janine's throat culture. Okay, this is our lab technician.